वीडियो में हम मल्टीपल एलील्स के बारे में पढ़ेंगे। तो चलिए शुरू करते हैं। जैसा कि आपको मालूम है, अल्टरनेट फॉर्म ऑफ जीन को हम अलील या अलीलोमा कहते हैं और मल्टीपल एलील्स की तारीफ हम इस तरह करते हैं। Allele existing more than two different forms are known as multiple alleles. Or we can also define it, three or more than three alleles are called multiple alleles. Or we can say that presence of more than two alleles of a gene is known as multiple alleles. Multiple alleles are located at same locus on homologous chromosomes. And we know that homologous chromosomes are those chromosomes which have their same shape, structure, and function. And locus means the position where the gene is present, where the allele is present. It is known as locus. Multiple allele arise due to mutation, or we can say that multiple allele arise due to gene mutation. For the study of multiple allele, population study is essential. In population can be defined a group of individuals of the same species live in an area in a particular time is known as population. In a diploid individual, study of multiple alleles is not possible because a diploid individual has only maximum two alleles of a gene. As we know that multiple alleles arise due to gene mutation. So some genes may have as many as 300 alleles. Here is a question is arise, but individuals have only two of these alleles. Why? So the answer is that because individuals have only two biological parents, we inherit half of our gene alleles from mothers and the other half from father. So we end up with two alleles for every trait in our phenotype. So in a diploid individual, study of multiple alleles is not possible because a diploid individual has only maximum two alleles of a gene. In case of multiple allele, number of allele in diploid individual is two, as they have two biological parents, one is father and the other is mother. And if gametogenesis are taking place, spermatogenesis or oogenesis, within the gametes, mean the egg and the sperm, the allele is a 1. In population, all types of allele should be possible. So the types of genotype can be calculated by this formula that n is equal to n plus 1 divided by 2. And n is equal to number of alleles. So one genes, they may either be completely dominant, incomplete dominant, codominant, or poor dominant, as we will discuss it in the other video. Individual one have either each type of their alleles. So two alleles, they have three genotypes, and three alleles, they have six different types of genotype when we study it within the population. You clearly see here in the picture that two alleles have three genotypes, while three alleles have six genotypes. So in individual one and individual two. The best example of multiple allele is ABO blood group system. As we know that ABO blood group system is discovered by Karl Landsteiner at University of Vienna in 1901 while studying blood transfusion in humans. That why sometimes blood transfusion it causes death of the human being and sometimes it saves the life. So Karl Landsteiner in 1930 received the Nobel Prize for this for the ABO blood group system discovery. As we know that ABO blood group system is the best example of multiple alleles. So blood group is controlled by I gene, and this gene located on chromosome number 9. I genes regulate attachment of sugar molecule on the surface of red blood cells. Or we can say within the I genes, the mutation takes place in three alleles are produced. That is IA, IB, and I naught. So IA and IB is completely dominant over the I naught, and IA and IB alleles are co-dominant with one another. Remember it. So concept to understand, a person having blood group A. So gene allele A is present in their chromosome. It produces the enzyme A, and this enzyme A is responsible to produce nitrogen, acetyl galactosamine. and it produces the antigen A, and the blood group is called blood group A. And a person having blood group B, having allele IB in their chromosome, it produces the enzyme B, and enzyme B produces galactose. So it produces the antigen B in blood. The person blood group will be B. And a person having blood group O, it does not have any alleles in their chromosomes. So enzyme is not produced and antigen is absent. So the person blood group have O. So we can easily conclude it here that a person having antigen A on the surface of red blood cells, it have blood group A, and a person having antigen B on the surface of red blood cells, it have B blood group, and a person having neither of antigen on the surface of red blood cells, it is O blood group, and a person having both type of antigen, antigen A and antigen B on the surface of red blood cells, their blood group will be AB. So based on the presence and absence of antigens on the surface of red blood cell, there are four different types of blood group: A blood group, B blood group, AB blood group, and O blood group. And a person having, as I told that a person having blood group AB, it has both type of antigen, antigen A as well as antigen B. So the alleles, or we can say that the genotypic study of a blood group is studied by Bernstein, which is a student of Karl Landsteiner. In 1925, he studied the genotypes of a blood group. So the phenotype of blood group, there are four types: blood group A, blood group B, and blood group AB, and blood group O. And their genotype is given within the table that blood group may either be homozygous or heterozygous, blood group B may either be heterozygous or homozygous, and blood group AB as well as blood group O. So blood group A are those blood group in which the antigen A is present. B blood group are those blood group in which the antigen B is present. And blood group AB are those blood group in which antigen A and B both are present. And blood group O are those blood group in which no antigen is present. Here, blood group A is called universal donor because in their red blood cells no antigen is present, so it gives the blood to all types of blood group. While AB blood group having both type of antigen on their red blood cells, so it receive any type of blood group from the person. So an ABO blood group system can be summarized. It have a single gene which is known as I gene, which is located on chromosome number 9. It have three alleles IA, IB, and I0, and it have six phenotypes and four phenotype A, B, AB, and O. Dominant blood group A and B is completely dominant, or we can say that IA and IB is completely dominant over the I not. IA and IB is co-dominant with one another, so it have multiple allele, and incomplete dominance is not apply in the multiple allele. Now we will go to take some examples to understand the multiple allele very easily. Father is heterozygous for A blood group and blood group of mother is AB. How many types of genotype and phenotype are possible in this class? So we take the example of father which is heterozygous and having blood group A. So the genotype of a father is IA, I0, and the mother genotype is IA and IB. You can see in the Punnett square or in the checkerboard that it has three phenotypes. So blood group A, blood group AB, and blood group B. And it has four genotypes. A child blood group is O. His parents' blood group cannot be. B and O, it produce the blood group O. A and O, it produce the blood group O. And AB does not produce the blood group O. So, and AB also produce the blood group O. So, all 1, 2, and 4, it produce the blood group O, and AB does not produce the child having blood group O. What will be the phenotype and genotype of a parental blood group of a family in which all four types of blood groups are present in their children? So it is a heterozygous blood group A and heterozygous blood group B. When a person having blood group A marries a woman having blood group, but both are heterozygous, so then four types of blood group will be possible in their children, either in their daughter or either in their son. A, B, AB, and O blood group. A man of a man of A blood group may be a woman of AB blood group. Which type of blood group in this child would indicate that the man is heterozygous for A blood group? As we know that we take two cases here. Case one in which the main, we can say that the main blood group is homozygous. So if in case one, when we take the example of homozygous, if it cross with a female blood group AB, so A and B blood group is possible. While in case two, if the male is heterozygous for A blood group in woman blood group have AB, so then A, AB and B blood group. When B blood group is produced, so we can clearly uh, we can clear that that the main blood group is a heterozygous. If you like the video, please share, subscribe, and watch.